Let's try. Hello everyone. I'm happy to be here again and that everyone is here also. I'm going to quickly present my work about isolating and characterizing cyanobacteria for plant growth promotion and stress tolerance. This work's divided in the introduction and the biology of cyanobacteria. Then I'll talk about their relevance in agriculture and when I had to actually isolate and test them in plants. I worked at this laboratory that studies heterotrophic plant growth promoting bacteria from Portugal, Cape Verde, and Angola, primarily rhizobacteria and plants. Because I had already briefly worked with cyanobacteria by extracting pigment and analyzing their DNA, and because I found the topic of using microorganisms as fertilizers important, I chose to mix the two things because cyanobacteria can help plants by producing herbicidal and insecticidal compounds, by solubilizing nutrients and providing nutrients for them, and because they are cost effective. A little about cyanobacteria is that they were the first oxygen producers. They photosynthesize with chlorophyll A like plants and their chloroplasts, unlike other prokaryotes. They are most related to chloroplasts in higher plants and purple bacteria. They have unicellular and filamentous forms in many different sizes, and they exist in most environments, a success that is due to their resilience to variations in light, salinity, pH, temperature, because they have a higher temperature optima than most eukaryotic algae, and because they display symbiotic relationships across all kingdoms. Here is how they might be divided in groups. We can see that some groups have traits like nitrogen fixing and filamentous forms, heterocyst forming capacity, branching filaments, and multiple fission. For their membrane, they are gram-negative, but have a much thicker peptidoglycan layer than most gram-negative bacteria. Some possess a mucilaginous sheath, and these might have evolved due to early Earth's high ultraviolet radiation. For their physiology, they use sunlight and water as electron donors for photosynthesis. But in the presence of light, they can still use organic compounds as carbon sources or switch from oxygenic to anoxygenic photosynthesis when sulfide concentrations are high, producing sulfur. In the absence of light, they can also, some species do fermentation or aerobic respiration of carbon reserves. The photosynthesis occurs in chilacoids, which are membranes in the cytosol with chlorophyll A and accessory pigments. These pigments are the soluble phycobilisomes, and they include the phycoerythrin, phycocyanin, and allophycocyanin. Each one harvests a different part of the solar spectrum, but uh, unlike many other phytoplanktons, they enable cyanobacteria to grow under low light. We can see that they are capable of producing many colors due to the different ratio that they acclimatize with these pigments. For survival and dispersal, many move by gliding towards light or chemicals. Many possess gas vesicles to regulate buoyancy and light exposure. And filamentous species can produce specialized cells like Ormogonia to facilitate colonization, akinets, which are spore-like resting cells, and heterocytes, which help with fixing nitrogen and slow oxygen diffusion to do that. How cyanobacteria help plant growth promotion is by promoting cell division and plant elongation. They can also suppress weeds. They enrich the soil, have many symbioses with plants, and improve soil aggregation, water infiltration, and rotation. Nitrogen fixation is the most reported plant growth promotion mechanism. It is catalyzed by the oxygen-sensitive nitrogenase, making unicellular cyanobacteria fix it only at night typically, and filamentous species often differentiate heterocytes. For the azopolysaccharide or EPS production, it's another plant growth promotion trait. They are high molecular weight carbohydrate polymers that envelop cyanobacteria. They improve soil structure and increase organic matter content, enhance phosphate solubilization by maintaining optimal pH for organic acids, and provide energy for heterotrophic microorganisms that might also benefit the plant. For the phytohormone production, the most common being the indolate-free acetic acid or EAA. AEE. I struggle to say that, I don't know why. It enhances seed germination and plant development, boosts physiological processes like photosynthesis and gas exchange, and phenolic acid and flavonoid production, stimulates the growth of beneficial soil microorganisms, and increases soil quality and crop yield. Phosphate solubilization is another thing they can do because after nitrogen, phosphate is the most critical for plant growth. Soil often contains a lot of phosphate, but only a very small fraction is available for plants without solubilization. Solubilization occurs by various mechanisms, such as the formation of EPS, chelation in acidification, and enzymatic cleavage. As for the alleviation of plant abiotic stress, when under drought, cyanobacteria have been shown to promote germination and absorb and eliminate heavy metals. Under intense heat, they might reduce oxidative stress and improve crop resistance to heat and drought. And under salinity, EPS can help a lot to mitigate it. And they can also improve growth and chlorophyll content and nutrient intake in plants, and accelerate growth and increase water content. So now is the practical part of this work. My objectives were isolating, culturing, and characterizing cyanobacteria from soil and water, then to access their plant growth promoting traits, and then to evaluate their ability to enhance plant growth and tolerance to abiotic stress. For the sampling of superficial water and soil, I went to freshwater or saltwater sites with falcon tubes, tweezers, and a grabber tool and a cooler. And I got in total 31 samples from 7 locations. I went to Rio de Janeiro, a fish pond, 3 lakes, a channel of the Boco River, and even got some samples from a plastic drinker for chickens. To isolate these strains, I initially transferred them to BG11 agar, that's medium made for cyanobacteria and the most common literature for them, and incubated them for up to 12 days. For each sample's water, I used a glass spreader, and when there was only soil or something looked cyanobacterial in the soil, I used tweezers and plated on separate plates that I labeled solid matter. Then the subculturing was performed in optimal colony forming unities exhibiting a blue-green tone, though they might have many other colors. I also attempted to make enriched strains, which I took a little of these colonies and let sink a bit longer in BG11, hoping to favor cyanobacteria in there. And I continued streaking optimal cultures this way up to 5 times per isolate after incubating for 3 to 10 days, hoping to avoid other contaminants. To maintain cultures, I used a range for a branch a large group of cyanobacteria, which is room temperature 16 to 8 or light dark cycle in an illuminated shelf. Um, then I assessed then under the microscope. It was a Zeiss Axioplan 2 imaging microscope at 100 magnification and an Olympus DP70 camera. I conducted this on 2 separate occasions with different cultures that were presumed to be clonal. Then I tested them in vitro. For the assays, I did culture media. For the nitrogen fixation, I used Ashby's nitrogen-free medium. For the siderophore production assessment, Chrome Azro S. For phosphate solubilization, modified Pikovskaya, potassium Alexandrov medium, and EAA reduction, I used the East mannitol broth with the Sakowski reagent method. Our plates were incubated at room temperature for 10 days, and then colonies or the halo that they created was measured in centimeters. For the siderophores assay, with siderophores being iron chelators that enhance iron uptake in plants, the assay relies on a shifting color from blue to orange, which means that the microorganisms that grew in there were able to deplete it from the medium, making this orange halo that can be measured. The phosphate solubilization assay is just like it, but then they create clear halos. The potassium is the same. The nitrogen fixation was assessed by the simple growth of the cyanobacteria in the medium because it lacks nitrogen sources. To quantify EAA, AEE, we supplement cultures with tryptophan, which is a primary precursor for bacterial AEE synthesis. Then we add the Sakowski reagent, which reacts with indole compounds and produces a pink color that is proportional to the phytohormone concentration, which can then be have its absorbance measured and having a standard curve of it made. For the plant growth experiment, I used sandy soil in 2 planting trays that I divided in 3 groups. The salinity stress tray was to get plants that will receive 100 millimolar salt sea irrigation. The drought stress tray would receive limited irrigation, and the no stress group was distributed across both to control for bias, receiving regular water. Each tray got 5 replicates of 4 cyanobacterial inoculants and 1 uninoculated control. I germinated cherry tomato seeds on agar after sterilizing them for 3 days under the same conditions that I plated cyanobacteria. When I transferred them to trays, I had already put on the isolates, cyanobacterial isolates, which had 60 replicates each in 5 milliliter PG11. I vortexed them and pooled and create solutions that got 4 milliliters in each tray cell. And for the controls, they got 4 milliliters of water instead. For the biomass and pigment quantification, I took the plants out, washed the roots, and weighted the shoot and root separately. Because they were very small, I made technical replicates per condition, pooling them to uh, analyze chlorophyll A and B and carotenoid content. I did this by taking the aerial tissue, macerating it in 80% acetone, centrifuging, getting the supernatant, and measuring the absorbance of that at the specific ratios for these pigments. Results and discussion. I managed to isolate cyanobacteria from two freshwater sites, which were the Perrapado Lake and Calvon Lake, and the plastic drinker for chickens. The initial plating yield mixed cultures dominated by green algae and heterotrophic bacteria, because cyanobacteria often grow slower than them. Therefore, among my many samples, only 6 isolates were confirmed via microscope to contain cyanobacteria. To characterize these strains, I saw that all of them were from the genus Lyngbia order Acelatoriales, again using a microscope, because they are filamentous, they have no branches, no actinids or pterocytes, meaning that they probably glide across surfaces. They have EPS, and their pigmentation varies a lot from dark green to pale blue-green. This is an image of the Calvon Lake isolate with 4 bright green Synedesmus cells, which is a green algae. That's a small amount of contamination that I found. These are the other samples that were used in the plant growth experiment. For the nitrogen fixation results, all of them managed to grow, confirming a diazotrophic capability, with P5 having the highest average diameter and E2 the lowest. These differences may reflect different nitrogenase efficiency or metabolic trade-offs, and fixation might have occurred under microaerobic conditions or at night, because these cells do not have heterocytes. For the phosphate and potassium solubilization, no halos were found, meaning that the cyanobacteria either were incapable of solubilizing that, or the assays had suboptimal conditions. For the siderophore, each isolated managed to produce a halo, with the highest diameters being C and P3, and the lowest E2 and E. May reflect strain-specific adaptations to iron availability in their native environments. For the EE prediction, the highest values were E2 and P5, and the lowest C and E. Again, this might indicate that these strains are different in somehow, might be due to their difference in tryptophan uptake, so the test needs to be improved, or their genetic efficiency to produce phytohormones. For the biomass, under no stress, we can see that P3 influenced the plants the most, greatly increasing their weight, both in shoot and root. Under drought stress, all of them, for having received cyanobacteria, got much lighter plants. And under salinity, we can see that P5 made them much heavier in shoot and root, perhaps due to its superior AEE production, have mitigated the damage from the salt by enhancing root expansion. For the pigment content, some interesting things to note here is that for the isolate E, shoots under drought had the lowest biomass, but more pigment than controls. possibly because of E's thick EPS delaying plant water and reducing oxidative damage. Under salinity, P5 showed less pigment than controls, suggesting that the salt disrupted its thiccoid membranes, and P3, in comparison, maintained moderate pigment under salinity, again indicating strain-specific resilience. In conclusion, no strain excelled universally. The stress alleviation was context dependent, but despite methodological constraints, the isolates showed potential as biofertilizers, with P3 being good for general agricultural use because it has a balanced siderophore and AEE production. E showed a lot of drought tolerance, which could suit water limited regions, and P5 could help plants in saline soils when paired perhaps with halo-tolerant bacteria to offset its limitations. Future research should prioritize genomic analysis of stress-responsive genes, field trials, and culturing optimization. Thank you.